Hi, my name is Glucose. Many people have been doing a Draw My Life video, so I decided to do one myself. Let's start off with my family. My grandfather was a carbohydrate. When he broke down, he created my mother, Starch. After my mother broke down, I was created, Glucose. I'm composed of 6 carbons, 12 hydrogens, and 6 oxygens. My life starts at the beginning of cellular respiration, a process humans use to get energy, otherwise known as ATP. I go through a total of 5 phases, the first being glycolysis, a process to cut me in two. In the cytoplasm, I meet up with 2 ATPs, or adenosine triphosphates. In order to split me in half, each ATP has to break off one of their phosphates and attach them to me, creating 2 ADPs. This process is called phosphorylation. Phosphorylation makes me unbalanced and able to split. The splitting process is known as lysis, hence the name glycolysis, the splitting of glucose. The 2 halves of me plus the phosphates are known as G3Ps. 4 ADPs then come collect the 2 phosphates to form 4 ATPs. NAD+ comes and collects the extra hydrogen ions, finally creating 2 pyruvates. After painfully being split in two, I have to go through link reaction. Let's focus on one half of me. My pyruvate meets with a coenzyme or a carrier. 2 of the carbon bond together, but my 3rd carbon gets thrown out. This is known as decarboxylation. Let's zoom into this particular carbon. Now that the carbon has no use in this form, its hydrogen ions are collected by NAD+ to form NADH+H+. The leftover carbon and oxygen combines with another oxygen molecule to form carbon dioxide. The purpose of link reaction is to use the coenzymes to move the 2C molecules from the cytoplasm to the matrix of the mitochondria. Remember, this process happens twice for both pyruvates. In my teenage years, I went through the Krebs cycle. First, the combined coenzyme and 2C molecule known as acetyl acid breaks apart. The coenzyme moves back to the cytoplasm and the 2C molecule stays in the matrix. The 2C molecule then combines with the 4C molecule to form a 6C molecule. Remember oxidative decarboxylation? One of the carbon breaks off and gives its hydrogen ions to NAD+ and forms CO2 with another oxygen molecule. Oxidative decarboxylation happens twice, from 6 to 5 to 5 to 4C. The 4C then rearranges itself. During this process, NAD+ comes and collects more hydrogen ions. Its friend FAD, an electron carrier, takes away hydrogen ions and electrons. At the same time, 1 ADP combines with a phosphate to form 1 ATP, or energy. But this whole Krebs cycle process happens twice. This results in 2 ATPs, 6 NADH+H+, 2 FADH2s, leaving me as 2 4C molecules. I then go back into the Krebs cycle to help my siblings remove their hydrogen ions and electrons. The other parts of me, like my hydrogen ions and my electrons, go through the next phase, electron transport chain. This occurs around the inner membrane of the mitochondria. First, NAD+ comes and drops off hydrogen ions. Some of the ions travel through the channel protein to the intermembrane space. Then electrons travel through the inner membrane, providing energy to push the rest of the hydrogen ions to the other side. Next step, oxidative phosphorylation. The hydrogen ions travel until they find an ATP synthase. They go through chemiosmosis, traveling back to the matrix. Let's zoom in. Chemiosmosis powers phosphorylation, forming 32 or 34 ATPs. Hydrogen's friend oxygen comes along, and the electrons came rushing late, making the oxygen molecule split in two. Finally, the oxygen and the hydrogens bond to form water. And that's my life as a glucose. See you later!